We can start, Dr. Fatma.
Good evening, everyone. Magandang gabi po sa inyong lahat. So let us start tonight's event with a prayer. Let us all put ourselves in the presence of God. Bismillahirrahmanirrahim. In the name of God, the most merciful, dear Almighty, and our ever-loving God, we glorify and thank you. You have showered us with so much blessings. Your presence continuously reminds us of your faithfulness and guidance. We humbly ask you to shower our speakers today of your greatest inspiration so that they may share the most of their knowledge, heart, and soul to their respective topic. May we observe the invaluable knowledge and put it into practice what we may learn today. We pray that you bless all the committee in charge, that they may be able to fulfill their task responsibly, that the objectives they have set may all be achieved. Your infinite blessing would mean the success of this activity. May we be a living witness of your genuine love through the enactment of the knowledge acquired through this activity. We ask this in the name of our Lord and Savior. Amen.
Philippine Society of Diabetologists. PSD's mission: the pursuit of excellence in diabetology. PSD's vision: excellence in diabetology. PSD's objective: to keep abreast of the most recent developments in all fields of diabetes research, education, and treatment.
PSD Statement of Purpose. The purpose of PSD is to give persons with diabetes lives with qualities unaffected by the condition. This is their right more than a privilege. This passion sustains and empowers us to find meaning and fulfillment. The purpose of PSD is to understand and accept that diabetes is a complex condition affecting an individual, the family and community. Knowledge of its biomolecular and cellular origins and understanding all its interventions are as important as mastering its psychosocial dynamics. The purpose of PSD is to be a family of responsible persons bonded by one passion. Agreeing in disagreements strengthen our unity. Criticisms fuel our growth. Love fills our camaraderie. The purpose of PSD is to be a source of inspiration that informs, guides, and corrects one another in daily practice. We find happiness in sharing knowledge and experiences that improve patient lives. The purpose of PSD is support individuals and organizations with similar noble goals, acknowledging that we all live in one world and fighting diabetes together multiplies good outcomes with a fraction of the effort. Collaboration drives inspiration, motivation and creativity. The purpose of PSD is to welcome the cure. When diabetes no longer exists, we will reincarnate with new passion and direction. Our vision will evolve with time, but the family lives on.
Okay, so again, this is Dr. Fatma Tiu. I am the current auditor of the Board of Directors of the Philippine Society of Diabetologists for the year 2020 to 2021. So in behalf of the officers and members of the Philippine Society of Diabetologists, under our President, Dr. Elwin Fernando, let me formally welcome you all to tonight's event. So this special edition lectures by the Philippine Society of Diabetologists, as you all know, is taken an activity taken by the Board of Directors to enhance our knowledge in pursuit of excellence in diabetology during this time of pandemic caused by the COVID-19 virus. So in line with this, we are having this special edition lectures, and these lectures hope to keep our alumni abreast of the informations in diabetes. So I would like now to go on by presenting to you our objectives for this evening. Our topic will be DM basics and its complications. So our objective will be to discuss the updates on the pathophysiology and the management of microvascular and macrovascular complications of diabetes. So our program will be in two parts. The first part will be a lecture in partnership by our in partnership with Sanofi Aventis. It's a CME grant. Our speaker will talk about the fresh perspective on glycemic control using a second generation insulin, after which it will be followed by another lecture. The topic is Biochem of Insulin Action by Dr. Marivic Villamor. So let us now start with tonight's event. The first part, which is again a CME grant from Sanofi Adventist. And let me now introduce our speaker for tonight. He had his Doctor of Medicine from FEU. He is known to us. We know him very well. He is a faculty of the UERM, Institute for Studies on Diabetes. He is the current head of the ISDFI Research Unit, and he is the past president of the Philippine Society of Diabetologists in 2017 to 2018. He also is an active consultant at the Cardinal Santos Medical Center and a visiting consultant at the Pasay General Hospital. So, friends and colleagues, let's all welcome Dr. Ernesto Ang. Good evening po, sir.
Hi, everyone, and thank you for that nice introduction. And I would like to take this opportunity to thank the Society, the Philippine Society of Diabetologists, for giving me this opportunity to present the Sanofi's module in this PSD Select. Okay. I was tasked by the company to deliver a presentation entitled Celebrating Legacies, Fresh Perspectives on Glycemic Control Using Second Generation Insulin. This is in time for the 100 years of insulin discovery. As you all know, in 2021 marks the 100th anniversary of discovery of insulin. So before everything else, I would like to give my disclosure, and this is my disclosure before the pandemic and during pandemic. And you can see it's depressingly short. So since this is sponsored by Sanofi, I'm obligated to say favorable things for the company, but I will try my best to be as balanced as can be. So let's put this out of our way and go to the presentation. So let me start from the past. Okay, just a glimpse of the past about history of insulin. It was in 1921, nearly 100 years ago, when the first injection of insulin purified from animal pancreas was able to bring life to a young boy with diabetes. And this young boy was named Leonard Thompson. And this is the pictures before insulin and years after insulin. Okay. And as we celebrate this life-saving discovery, we marvel the amazing story of insulin and how it led us to a novel therapies for diabetes. Mr. Thompson showed signs of improved health and went on to live 13 more years, taking doses of insulin, eventually dying of pneumonia at age 26. Until insulin was made clinically available, a diagnosis of type 1 diabetes was a death sentence, more or less quickly, usually within months and frequently within weeks or days. Since then, development in insulin have come a long way. And we are proud to recognize the progress made in the insulin development over the last century and the advancement yet to come. Now, to better illustrate this, here is the sentimental journey of the major character in the discovery of insulin, beginning with Banting idea, um, his meeting with uh, MacLeod, and experiment and purification of the extract, and how the pharmaceutical companies become involved in the plans for mass production. Highlighted here, it is discovery uh, that is granted eventually with a Nobel Prize. Now, this is the two discoverers, Dr. Frederick Banting on your left, a Canadian surgeon, and Charles Best, a medical scientist student. Again, this is the co-discoverer of insulin. However, there are four persons that were recognized for the discovery of insulin. As I already mentioned, Banting and Best, and MacLeod, Professor John MacLeod, the chairman of physiology of University of Toronto, an expert in carbohydrate metabolism in his own right. His main contribution was he provided the much needed lab space for the experiment. While John Beltran um, Collip responsible for the purification of insulin. So Banting divided his share with Best, and MacLeod split his portion with Collips. Now, the discovery of insulin is indeed life-saving. However, as uh, can be seen in the fluctuation of the glucose level in the original patient chart, there was significant variability between insulin batches that made it difficult to determine the proper doses and avoid hypoglycemia. Now, we go now to the evolution of insulin. Okay, so from left to right, as you can see, the initial use of insulin is for survival of patients with type 1 diabetes. However, as we manage hyperglycemia, we often run into hypoglycemic trouble. Moreover, because of insulin crystallization, it requires multiple daily injections for it to be effective for 24 hours. In 1946, with the discovery of MPH, neutral protamine hagedorn, which protracted or prolonged the insulin injected parenterally, that reduces the number of injections. In 1982, the introduction of human insulin causing less antibodies, thus less allergic reaction. In 2000, the introduction of basal insulin analog given once daily was a welcome improvement. In 2015, the second generation basal insulin analog came along. So here is the spectrum of insulin development have come a long way from survival to management of hyperglycemia and minimizing hypoglycemic risk. Now, we fast forward to 2021. Approximately 4 million people died from diabetes. A total health expenditure on diabetes is estimated at a staggering 727 billion US dollars, And we know that diabetes complications can be prevented by good glycemic control. Now, let us discuss a little bit of type 2 DM pathogenesis. Here's the data of UKPDS, a landmark study for type 2 diabetes. As we know about UKPDS population of diabetes, that they are relatively, the recruited population were relatively newly diagnosed, and they found out that at the time of diagnosis of this patient, they have already lost approximately 50% of their beta cell function. Now, Professor DeFronso's study at San Antonio, Texas patient, more recently, that's about 2 decades later, looking at disposition index, have shown much worse beta cell function. And by the time on this population, by the time that the diagnosis of type 2 diabetes is made, the patient had lost more than 80% of beta cell function. Now, this figure shows a ballpark figure on how A1C reduction we can expect from a different diabetes medication given as monotherapy. The top three performers for the oral medicine would be your SU, metformin, that's about 1.5 reduction of A1C, okay? TCDs and SU. TCDs is about a maximum of 1.5, SU and metformin, but in comparison with insulin has a greater effectiveness at an A1C lowering at 2.5% or greater. Part of the reason is that unlike your other pharmacologic diabetes therapies, especially oral, insulin has no dose limit. Now, many type 2 DM patients will require injectable medication within 5 to 10 years of diagnosis, and this is because of the natural history of type 2 DM. Type 2 DM, as we know, is a progressive disease, and the progression may not be related to the medication being used. In addition of insulin to oral medication is well accepted and more effective with less hypoglycemia and weight gain for basal insulin versus other insulin regimen. The recommendation in initiating injectable therapies in patients uncontrolled with oral anti-diabetic drugs. Now, if the A1C is above target, okay, despite dual or triple therapy, consider GLP-1 receptor agonist in most prior to insulin. Now, why? Because it lowers risk. It has a lower risk of hypoglycemia. It is associated with weight loss and a significant lowering of A1C. If above A1C target after 3 months, if the target is not reached, then you can add basal insulin. Okay, but choose a basal insulin with a lower risk of hypoglycemia and lower glycemic variability. Now, there's another choice. You can consider insulin as a first injectable if the A1C is very high, like 11% or higher than 11%, and patient usually have the symptoms or evidence of catabolism, and if you are considering that the patient uh, may be a possibility of a type 1 DM. Okay, so the question is why basal insulin? Okay, treating fasting hyperglycemia lowers the entire 24 hours plasma glucose profile with or without food. Besides, patient will accept the regimen once daily injection more than multiple daily injection, and it simplified the blood glucose monitor. Let's focus our attention on these slides. Consider blood glucose profile of an uncontrolled diabetes in orange curve on the upper level. The fasting plasma glucose is 200 plus, that is in orange. Definitely, this is hyperglycemia due to an increase in fasting glucose. And you can notice Following a meal, the PPG or postprandial glucose becomes even more higher. That's more than 300. So if you bring down the FPG to 100 by basal insulin, will also successfully bring down the PPG that is curved, that is in the yellow curve. Okay, so Monier et al. In 2003 had given us the evidence that both FPG and PPG contribute to A1C. And the relative contribution of PPG or postprandial glucose excursion is predominantly in fairly controlled patients, whereas the contribution of fasting hyperglycemia increases gradually with diabetes worsening, meaning the higher the A1C, the higher the contribution of fasting plasma glucose. As you can see on your right side of the graph, you see the fasting plasma glucose in light blue, higher A1C has a higher fasting plasma glucose. Now, more recently, studies in 2017 among Asian population, this was validated. The Monier study on your left, this is the Malaysian population or data. You see again, the higher the A1C, the higher the fasting. And on the right, the Korean patient or the Korean data, the same thing. So in this study, fasting hyperglycemia was predominantly higher if the A1C is higher. Therefore, we focus the therapy on our fasting plasma glucose. So FPG is a determinant of PPG excursion. So the dictum is to fix the fasting first. Okay. And this is one study to prove the concept. Lowering fasting plasma using glargine as a monotherapy over a 9-point SMPG, there's no statistical difference between premixed co-formulation of basal postprandial insulin. As you can see, the a light blue curve with co-formulation versus insulin glargine, and you can see that during the 52 weeks period, both treatments show similar mean 9-point SMPG profile throughout the meal. However, significant lower pre-breakfast fasting SMPG were observed with IGLAR versus the co-formulation mixture of basal and prandial insulin. Okay, so effective therapy requires balance between benefit and glycemic control and risk of hypoglycemia. Timely, effective, stable glycemic control improve A1C, prevention of complications, less restrictive regimen may improve adherence as well as quality of life, low risk of hypoglycemia, may allow optimal dose titration and improve glycemic control, may improve adherence, may result in reduced mobility and healthcare resource utilization. Now, we go now to the new consideration on glycemic metrics. So, as I already discussed in the previous slides, the discovery of the use of commercial insulin was for survival, especially in type 1 DM, then improvement in the purification, stabilization, and prolonging or protecting the effect of insulin, and this is to control the peaks and dips of blood glucose. In 1970s enters the blood glucose monitor technology. In 1976, the first home use of self-monitor blood glucose or SMPG. In 1999, the advent of continuous glucose monitor or CGM, and in 2013, the standardization of CGM reporting, and so the so-called AGP or the ambulatory glucose profile. Now, this slide shows you the advantages and limitation of HbA1c. Okay, the advantages is easy to measure and standardize, relatively cheap, predictive of vascular complication because this is used in DCCT as well as UKPDS, often used to guide management in the decision. However, there is a limitation, and limitation are as follows. A1C is a poor indicator for glycemic variability, potential confounding by some condition like renal failure, hemoglobinopathies, etc., and only provide an average measure of glycemia. And therefore, there is a need for a metric beyond an A1C to address these limitations. Now, this is the new glycemic metrics, an example of AGP or the ambulatory glucose profile. The AGP shows glucose pattern over time, which provide considerable information for optimizing diabetes management by identifying specific times of day with hyperglycemia or hypoglycemia. There are numerous studies have shown that there are a wide range of possible mean blood glucose level for a given A1C level, meaning for some patients, A1C may not be a reliable indicator of glucose control. In addition, A1C primarily reflects hyperglycemia and does not provide information about hypoglycemia, glycemic variability, or the daily pattern of glucose concentration. In other words, do not reflect the fluctuation of blood glucose and may not provide enough information on how to adjust the treatment regimen right away. So a better determination is continuous glucose monitoring, CGM, and it display all the report a variety of metrics and targets along with a 14-day composite glucose profile as an important aid to clinical decision making. This metric used to for addressing or assessing hyperglycemia, hypoglycemia, and glycemic variability. This daily profile can inform an immediate therapy. And this is an example. There are several data that are included here. Number one, you have the dates. Number two, the percent time of CGM that is active, that will give you a percentage, the glucose ranges, the average glucose, and the glucose management indicator, which is calculated from the average glucose, which is the estimate for your future A1C, and of course the glucose variability. And the lower the glucose variability, the better. Okay, and on your right, you can show you a graph like your target range, the target range or target in range or TIR, which is pegged as 70 to 180. They'll give you a percentage, okay, the green one, and the yellow is high, and the orange is very high. And they can also give you the lower or the hypoglycemic value, low and very low. So this is the green, and more green, better, and we want less red. Okay, we want more target range, and we want less hypoglycemia, especially severe hypoglycemia of less than 54 milligram per deciliter. So it also provides a graph that shows very high, target range, low, and very low. The TIR, ideally, the glucose values are in target range, meaning time in range defined as a percentage of glucose reading and time spent in the range within 70 to 100 milligrams per deciliter. The higher the TIR, the lower the future A1C level and vice versa. Okay, so there are data to show that TIR correlates well with the long-term diabetes complication. Okay, again, what's the relationship of TIR with A1C? Number one, A TIR of 70% corresponds with an A1C of approximately 6.7 to 7%. An increase in TIR of 10% corresponds to a decrease in A1C of approximately 0.5 to 0.8%. For each 10% reduction in TIR, there is risk of developing retinopathy, an increase by 64%, and microalbuminuria by 40%. So it can also give us a glycemic variability, and we all know glycemic variability can be a major barrier to the optimization of glucose control. And this is captured also in CGM, and this is not captured in A1C. Clinically, glucose variability is implicated in the diabetes complication, especially cardiovascular disease complication. So it needs to be identified and managed properly. Again, you can see the daily, peaks and dips. Okay, they can identify the glycemic variability. Okay, and this is captured again with the CGM. Okay, talking about how to avoid glycemic variability. The secondary generation basal insulin analogs, such as glargine 300, offer lower degree of variability. As you can see here, using glargine 300 versus glargine 100, you see that you significantly lower glucose variability when you use glargine 300. Glycemic variability is not only the spike of blood glucose, but also the dips, no? The hypos. No? And hypoglycemia, especially severe, is associated with the risk, increased risk of adverse clinical outcome or even death. So glargine 300 in this slide showed reduced hypoglycemia versus glargine 100 during titration period, as shown in this patient on your right, or rather on your left, patient level meta-analysis, and in the addition study on the right, showing lesser nocturnal as well as all-day hypoglycemia versus glargine 100. So the effective therapy requires a balance between the benefit of glycemic control and the risk of hypoglycemia. Timely, effective, and stable glycemic control. It improves achievement of A1C target, lower glycemic variability, and longer time within the range. Prevention of complication, lower health care utilization. Less restrictive regimen may improve adherence as well as quality of life. Low risk of hypoglycemia, reduce fear of hypo, may allow for optimal dose titration and improve glycemic control, may improve adherence, may result in reduced morbidity and healthcare resource utilization. So finally, the key message and summary. Fundamentals, principles of therapy are still relevant today. Insulin is still the single most effective glucose-lowering drugs. Fix fasting first. The principle is still relevant in current practice, reaffirmed by more recent data. Treating fasting hyperglycemia lowers the entire 24 hours plasma glucose profile. A1C remains to be the relevant gold standard or relevant parameter to assess glycemic control and outcomes. But new glycemic metrics such as TIR and glucose variability are not meant to replace A1C but to complement it. Insulin with lower glycemic variability and longer time within normal range help reduce morbidity and adverse clinical outcome. And with that, I would like to close my presentation and thank you for listening. But before I totally close and end, since most of us are feeling the pandemic and quite depressed, let me just give you a joke as an amusement to alleviate our depression, okay, somehow. And it's all about the words complete and finish, or the difference between the two, okay. So in there's no English dictionary has been able to explain the difference between the two words. Well, some people say there is no difference, but for me there is. And the difference lies here. Okay, when you marry the right woman, you are complete. But when you marry the wrong woman, you are finished. And if you marry a wife who likes shopping, you are completely finished. Thank you for listening.
Thank you very much, Dr. Ang. I'm glad my husband is not here listening to us tonight, as he may consider himself completely finished. So again, sir, thank you and congratulations for a very comprehensive and excellent presentation. So as Dr. Ang has mentioned, this year, 2021, we are celebrating the 100 years of insulin discovery. And we can say that this discovery of insulin in 1921 has transformed diabetes from a death sentence to a chronic condition that we know today. So insulin have since saved millions of lives and is considered to be one of the greatest medical achievements of all times. So to celebrate, we in the PSD join the rest of the world in the campaign to this celebration through the celebration of the many advances that has been done in the past 100 years, which have already transformed the lives of many people with diabetes, including those that we see in our clinics. So let us call on more action to ensure that all people living with diabetes have the best possible quality of life and health. So with this, we have invited an esteemed speaker who will bring us to the basics of insulin. She is a doctor of medicine from the same school where me and Kuya came from, the UERM, and where our ISD is part of this school. So she has a PhD in education, in research and education at the UP College, and had her master's in health professions education also in UP Manila. She has a diploma in community nutrition at the University of Indonesia, Jakarta, wherein she topped as a class valedictorian. She is currently a professor at the CEU Manila School of Medicine, MS2 Department of Education, Training and Research at the Philippine Heart Center. She is a faculty the biochemistry research methods in UERM Graduate School. So colleagues, let's all welcome Dr. Marivic A. Villamor. Good evening, Doctora.
Good evening. I would like to begin with a note of gratitude to the members of the Philippine Society of Diabetologists for this invitation and the opportunity to revisit the biochemistry of insulin action. Since the isolation of insulin in 1921 by Banting et al. In Toronto, Canada, there has been no peptide hormone whose mechanism of action has been more intensively investigated. The two-chain structure of insulin was the first complete protein structure to be elucidated through the pioneering work of Fred Sanger in 1959. This is not only a result of the utility of this hormone as a model hormone system for peptide hormones, but also a reflection of the importance of insulin in human biology and medicine. I have no actual or potential conflict of interest in relation to this presentation. Metabolic homeostasis in humans is tightly regulated by complementary actions of several hormones, among which insulin and glucagon. The presentation shall cover the journey of insulin from its biosynthesis, glucose sensing mechanisms, secretion and export, insulin actions on key organs, and its degradation and clearance from the system. The metabolism of this hormone from the pancreatic beta cells is controlled by metabolic, endocrine, and paracrine regulatory mechanisms and is essential for the control of blood sugar levels. The deregulation of these mechanisms may lead to various pathologies, notably type 2 diabetes mellitus, which is a defect in the combined lesions of impaired insulin action and the loss of normal insulin response to blood sugar levels. This year, 2021, marks 100 years since the discovery of insulin, a hormone that plays a vital role in energy homeostasis. Historically, insulin has been associated with blood sugar, and true enough, insulin has profound effects on carbohydrate metabolism. Its discovery revolutionized therapy for diabetes and is a triumph of modern medical research. After its isolation in 1922, almost overnight, insulin rescued patients with severe cases of diabetes from a rapid decline in health and early demise. Therefore, a detailed knowledge and understanding about the processes that involves biosynthesis and secretion would be critical to our understanding of diabetes mellitus, a disease characterized by high blood glucose levels, and which results from insulin insufficiency in the body. The journey of the biochemistry of insulin begins in the pancreas. The cellular processes related to the biosynthesis of insulin via pre-proinsulin and proinsulin and their intracellular transport, sorting, proteolysis, and ultimate storage in secretory granules are remarkably well integrated, both topologically and biochemically. Insulin is a peptide hormone composed of 51 amino acids distributed between two chains, the A and B chains, containing 21 and 30 amino acids respectively. Connecting these two chains are disulfide bridges that contain cysteine residues. On the account of the protein nature of this hormone, the synthesis of insulin is directed by a gene located in chromosome 11. Insulin mRNA undergoes translation as a single polypeptide chain called pre-proinsulin. Upon its translocation into the endoplasmic reticulum, the pre-proinsulin is cleaved at its signal peptide, releasing proinsulin. Proinsulin is a single chain containing insulin's A and B chains in a continuous manner joined through a segment known as the C domain. At the site of each dibasic residue, a trypsin-like enzyme cleaves proinsulin, releasing a C-peptide and mature insulin. Until it is needed by the body, insulin is stored within glucose-regulated secretory granules as zinc hexamers. Insulin biosynthesis in this manner is generally rapid and efficient, with only 1 to 2% of the protein remaining as proinsulin within mature secretory granules. The major proteolytic cleavages required for the converting proinsulin to insulin are summarized in this next image. In early studies, it was shown that the joint action of pancreatic trypsin and carboxypeptidase B gives rise to the naturally occurring conversion of products, namely C-peptide and native insulin. However, it has been identified that there are now two key enzymes, namely protein convertase 1,3 and PC2, that participate in proinsulin processing. In fact, Rhodes and co-workers were able to demonstrate the role of and order of the actions of PC2 and PC13 in pro-insulin conversion, where PC2 prefers the pro-insulin intermediate that has already been processed at the B-chain peptide junction as a substrate. Initially, PC13 acts to generate DES3132 intermediate, which is then cleaved at the C-peptide A junction by PC2. It has been suggested that the elevated accumulation of proinsulin and DES3132 proinsulin in diabetics might be due to a defective action of PC2. Unless there is metabolic requirements for insulin, newly formed insulin is stored in granules ready to be mobilized. Most insulin granules are stored within the beta cell cytoplasm at some distance away from the plasma membrane. The remainder move to the periphery along the microtubule networks in an AMPK kinesin 1 dependent mechanism. To reach the plasma membrane, however, granules must cross a cortical actin network that acts as a physical barrier to insulin secretion. The process is coordinated by the action of several small G proteins and their activating nucleotide exchange factors. Finally, secretory granules must duct at the plasma membrane and be chemically primed in response to intracellular calcium signals. The journey of insulin then proceeds to its secretion from the storage granules. The secretory process involves a sensing mechanism. Notably, beta cells can sense and respond quickly to postprandial elevations of blood glucose. The beta cells respond to changes in glucose concentration by varying the rate of synthesis and secretion. An increase in glucose concentrations above the threshold of 3 millimoles concentration in the pancreas stimulates B cells to secrete insulin in a dose-dependent manner with half maximal stimulation at 10 to 12 millimoles glucose concentration. Simple as it may sound, the succeeding steps in secretion of insulin are far more complex. The consensus model, as shown on the screen, reveals a relay of chemical to electrical to metabolic signals. This model highlights glucose as the paramount metabolic signal eliciting insulin secretion. Glucose is freely taken up into the beta cell via GLUT transporters mobilized to produce ATP, which then triggers a cascade of intracellular signals necessary for glucose-induced insulin secretion. The low-affinity GLUT2 facilitative glucose transporter functions in the beta cells of the pancreas to transport glucose in direct proportion to the circulating glucose concentration. This transporter functions in concert with glucokinase such that small increase in glucose influx signal changes in the ATP-ADP ratio, resulting in a highly sensitive mechanism regulating insulin secretion. While GLUT2 has been traditionally assumed as the major mediator of glucose uptake into beta cells, more recent studies in human islets suggest that other insulin-independent glucose transporters like GLUT1 and GLUT3 may play an important role. This redundancy may explain why certain individuals with variants in the gene encoding GLUT2, like Fanconi-Bickel syndrome, do not have significant abnormalities in insulin secretion. Once inside the beta cells, glucose would be readily phosphorylated by glucokinase to generate glucose 6-phosphate, which through glycolysis feeds the mitochondrial tricarboxylic acid cycle. This is the rate-limiting step of insulin secretion, and as such, glucokinase is considered the glucose sensor of the B cell. Once in the mitochondria, glucose 6-phosphate is metabolized by the Krebs cycle to produce ATP, The resultant ATP binds and closes the ATP-dependent potassium channel in the plasma membrane. The closure of this channel prevents potassium from leaving the beta cell, therefore resulting into the depolarization of the plasma membrane. Once the membrane is depolarized, the L-type gated channels are triggered, releasing influx of calcium into the beta cells. This will result in an increase in calcium intracellular calcium concentration that would eventually activate the peptide that will eventually activate the secretion of the pool of insulin-containing duct secretory granules and further stimulate the migration of the reserve pool towards the periphery. Glucokinase, therefore, can effectively generate downstream signaling metabolites like ATP and pyruvate within a range of glucose concentration that would match the normal physiologic range for plasma glucose homeostasis. For this reason, therefore, the tandem of GLUT1-2 and the glucokinase is often referred to as a glucose sensor controlling blood glucose levels. As previously discussed, we now appreciate the coordinated nature of glucose sensing. In fact, the pancreatic B cells act as a self-contained system to secrete insulin in response to blood glucose concentration changes. The priming of beta cells is similarly influenced by other nutrients, including free fatty acids, GLP-1, GIP, and enzymes like glucagon and adrenaline. The beta cells may also be similarly primed by incretins and parasympathetic neural input, even under fasting conditions. Now we have just traced the pathway or the journey of insulin from its biosynthesis to its secretion. But let us now pay more attention into the processes that are involved in the secretion of insulin from beta cells. Continuing with this journey, we attempt to describe the mechanism of secretion because under normal circumstances, the pancreatic beta cells retain a remarkable state where insulin secreted in response to a stimulus is rapidly replenished at the biosynthetic level in parallel, thus keeping intracellular insulin stores optimal. The vast majority of insulin, however, is secreted via the regulated secretory pathway and only occurs in response to increases in specific nutrients like hormones, neuropeptides, and some pharmacologic reagents. Glucose stimulated insulin secretion, or GSIS, is considered as the primary pathway for insulin secretion. It is augmented by amplifying pathways that include metabolic amplification by amino acids, free fatty acids, and even glucose itself. And second, this is amplified by neurohormonal molecules such as GLP-1 and parasympathetic and innervation. In general, most nutrients that regulate insulin secretion also control pro-insulin biosynthesis, and the most relevant physiologically is glucose. The first phase of the glucose-stimulated insulin secretion is a multi-step process that requires transport and oxidation of glucose, electrophysiological changes, and fusion of insulin-containing secretory vesicles with the plasma membrane. GSIS also shows a biphasic pattern. Shortly after glucose stimulus, the first burst of insulin secretion occurs, followed by a decrease in the rate of secretion, and then a second phase ensues. The second sustained phase of insulin secretion can be observed just after the decrease, which can continue for up to several hours until euglycemia has been achieved. There are several mechanisms that can account for this biphasic secretion, one of which is the pattern that reflects exocytosis of different functional pools of secretory granules. A readily releasable pool, or RRP, is a small fraction of granules that are thought to be pre-duct and prime at the cell membrane and thus immediately available for release upon stimulation. A reserve pool contains a larger amount of granules that sit farther beneath the surface of the plasma membrane, and they require trafficking and recruitment to the same release site in order to be secreted. In light of this model, the prevailing hypothesis is that the readily releasable pool is responsible for the first phase of secretion, and the replenishment of this pool from reserve pool is critical for sustaining a second phase of secretion. Although the mechanisms involved in the first phase of insulin secretion, termed the triggering pathway, are well understood, mechanisms regulating the sustained second phase or amplifying pathway are yet to be deciphered. Insulin secretion is a process that involves the fusion of insulin granules with the plasma membrane and exocytosis of granule content. Exocytosis is an essential cell biological process by which the contents of a secretory granule are released across the plasma membrane. The coordinated interaction of exocytic machinery proteins in association with calcium channels ensures assembly of an insulin granule exocytic site complex that is ready to go when needed. The priming process involving both granule modification and translocation toward the plasma membrane is the rate-limiting step for insulin exocytosis. Several observations suggest that there is a relationship between the biphasic insulin secretion and pools of granules. First, both the first phase of insulin secretion and the exocytosis from readily releasable pool can occur even in the absence of nutrients. However, both the second phase of insulin and readily releasable pool replacement are strictly metabolic product dependent. Another consideration pertains to the fact that the total number of granules in the readily releasable pool is positively related to the amount of insulin released in the first phase of secretion. We can therefore maintain that regulated exocytosis is a multi-step pathway, including trafficking, tethering, docking, priming, and fusion. In healthy subjects, insulin secretion is exact to meet the metabolic demand. Accurate control of insulin release is of critical importance. Too little insulin leads to diabetes, while an excess of insulin causes potentially fatal hypoglycemia. To sense a nutritional state, beta cells are clustered in islets that strategically connect to the vasculature. Islets form a dense network with small blood vessels and receive 10 times the amount of blood than cells in the surrounding exocrine regions. Interestingly, while glucose is the key physiologic stimulator of the insulin-secreting system, it can be replaced or enhanced by other energy-providing metabolites like leucine or succinyl-CoA that either feed into the glycolytic pathway or the Krebs cycle, or even related oxidative phosphorylation pathways. Insulin secretion is finely regulated by multiple mechanisms to ensure appropriate hormone delivery under changing short-term conditions like variable nutrient intake or long-term circumstances like obesity, insulin resistance, or pregnancy. Insulin secretion is a highly controlled process and is subject to a variety of regulatory mechanisms. I will pay attention in this presentation to the mechanisms of stimulated insulin secretion. There are many insulin secretagogues that induce insulin release. Some of these include glucose, fatty acids, amino acids, incretins, nucleotides, calcium, calcium-potassium electrochemical gradients, metabolic coupling factors or MCFs, and the cellular level of reactive oxygen or nitrogen species. However, glucose remains as the principal insulin stimulant as it is the primary source of food fuel. Nutrients promote insulin's exocytosis by a variety of mechanisms, including plasma membrane depolarization, by enhancement of the ATP-ADP ratio via glycolysis and the Krebs cycle, and the generation of reducing equivalents like NADH and FADH2, and the production of lipid signaling molecules that would promote vesicle trafficking and calcium influx. The response involves metabolic activation and generation of metabolic coupling factors that relay the nutrient signal throughout the cell and induce insulin biosynthesis and secretion. Glucose metabolism is central to the generation of metabolic coupling factors that may lead to insulin release, and the most notable of these MCFs is ATP. Now we have seen how insulin is released from the pancreatic beta cells. We now move to what happens to the key organs that will have the insulin receptors. Upon release from the beta cells, insulin would travel through the circulation to reach the liver first. Although many somatic cell types express insulin receptors, the role of insulin in glucose homeostasis is typified by insulin's direct effects on the skeletal muscle, liver, and white adipocytes. These tissues perform distinct roles in metabolic homeostasis, necessitating tissue-specific insulin signal transduction pathways. In other words, while they have the same insulin receptor isoforms, these three main organs would react to insulin in different biochemical patterns. In the liver, insulin activates glycogen synthesis, increases lipogenic gene expression, and decreases gluconeogenic gene expression. In the white adipose tissue, insulin suppresses lipolysis but increases glucose transport and storage of lipids. The actions, therefore, of insulin on the parenchyma of peripheral tissue are diverse, and paramount among them is the regulation of glucose metabolism. Insulin exerts all of its known physiological effects in a similar manner across all the different key organs. These organs contain the same insulin receptor known as your insulin receptor or INSR, which is located in the plasma membrane of the target cells. INSR is a heterotetrameric receptor tyrosine kinase composed of two extracellular alpha subunits and a transmembrane beta subunit. These are both linked by disulfide bonds, again containing cysteine residues. There are two isoforms of this receptor, the A and B isoforms. However, the B isoform is much more specific for insulin and is therefore expressed in the differentiated liver, muscle, and white adipose tissue. This isoform is thought to mediate most metabolic effects of insulin. Isoform A is more prominent during fetal development. INSR has two binding sites, but exhibits negative cooperativity, meaning that insulin binding at one site decreases insulin binding affinity in the other site. Binding of insulin, therefore, to the alpha subunit would induce a conformational change resulting in the autophosphorylation of a number of tyrosine kinase residues present in the beta subunit. Receptor activation would then lead to the phosphorylation of these key tyrosine residues on IRS proteins. The catalytic subunit of PI3 kinase then phosphorylates phosphatidyl inositol 4,5 biphosphate, leading to the formation of PTD345P3. A key downstream effector of PTD345P3 is AKT, which is recruited to the plasma membrane. Once active, AKT enters the cytoplasm where it leads to the phosphorylation or inactivation of key enzymes, thus resulting in metabolic signaling. This is a general pattern that can happen in all the key organs that possess this insulin receptor. Let's now focus on what happens in the liver. Insulin from the endocrine pancreas is secreted into the portal vein. So the liver is exposed to insulin concentrations 2 to 3-fold higher than those in the general circulation. The diverse anabolic actions of insulin are defined by the hepatic insulin signaling pathway. Hepatic insulin signaling starts with INSR, trans-autophosphorylation, activation, and recruitment of scaffold signaling proteins. The stimulation of net glycogen synthesis is therefore the major direct physiological function of postprandial insulin on the hepatocytes. Additionally, insulin rapidly and potently reduces hepatic glucose production by inhibiting the enzymes of gluconeogenesis. Insulin has also a direct hepatocellular effect on lipid metabolism. Most prominent among these effects is the transcriptional upregulation of several genes of de novo lipogenesis. The overall effect is to promote lipid storage in the hepatocytes and decrease the availability of fatty acids for oxidation by other tissues. Thus far, we have considered how insulin regulates the cellular synthesis of two major classes of biological macromolecules, lipids and glycogen. We now tackle what happens to proteins once insulin enters the hepatocytes. Insulin regulation of protein synthesis is largely mediated by signaling into the mammalian target of rapamycin or mTOR network. The principal direct actions of insulin on the liver, therefore, are to stimulate storage, thus it's called the anabolic hormone. Okay? And the storage would be as glycogen synthesis, which will be exported for the use of other tissues in case of need or metabolic demand. We have the storage of lipid through lipogenesis and counteracted by a decrease in the reactions leading to lipolysis. And finally, proteins are stored or formed in the hepatocytes, which can be used for other functions in other cells. We now move towards understanding or reviewing what happens when insulin binds to the receptors in another key organ, which is the skeletal muscle. The skeletal muscle is an energy-consuming tissue. Any energy the myocyte stores is mostly for its own use. With the exception of 3 carbon units like lactate and alanine, which are generated during glycolysis, and are eventually released by the skeletal muscle and mostly cycled back to the liver through the glucose-alanine cycle or the glucose-lactate cycle. Insulin signals the skeletal muscle that glucose is abundant. Accordingly, The myocyte insulin signaling pathway is specialized promoting glucose uptake and net glycogen synthesis. As mentioned, in contrast to the glycogen synthesized in the liver, the glycogen formed in the skeletal muscles will remain as the main energy source of the myocytes in turn when there are less concentration or less glucose levels in the blood. This is because of the absence of a hormone that will release glucose from glucose 6-phosphate in the skeletal muscle. Although both IRS1 and IRS2 are expressed in the skeletal muscle, the primary insulin receptor substrate in the muscle appears to be IRS1. Therefore, once glucose enters the myocyte upon stimulation by insulin, two possible fates happens to glucose. The first one would be that it's going to be oxidized through the glycolytic pathway, or it can now enter the glycogenesis pathway, resulting in storage of glycogen. The principal pathway of insulin-stimulated glucose disposal in both the healthy and type 2 diabetic human muscle is glycogen synthesis. Although acute insulin-stimulated increases in skeletal muscle glycolytic flux and glycogen synthesis are primarily a consequence of increased glucose transport activity in the muscle and subsequent allosteric regulation by glucose metabolites. Insulin independently regulates both glycolysis and glycogen synthesis. Insulin positively regulates the transcription of hexokinase 2, the primary skeletal muscle isoform of the first glycolytic enzyme, thus providing a slow control of glycolytic capacity. As the principal tissue for insulin-stimulated glucose disposal, therefore, skeletal muscle remains the primary driver of the whole body glycemic control. Skeletal muscles or the myocytes are uniquely responsive to muscle contraction or exercise with increased sensitivity to subsequent insulin stimulation. We now turn our attention to the insulin signaling involving the white adipocyte tissues. The white adipocyte is sensitive to insulin in vivo. The most critical physiological functions of insulin action in white adipose tissue are suppression of lipolysis and stimulation of glucose uptake. Insulin is the most potent anti-lipolytic hormone. The potency of insulin to control plasma non-esterified fatty acids is an important physiological function insulin in the white adipose tissue. Insulin suppression of plasma NEFA levels occurs through rapid inhibition of triglyceride lipolysis in adipocytes. The best understood mechanism for insulin suppression of lipolysis involves the attenuation or reversal of adrenergic signaling through CAMP and protein kinase A. Suppression of lipolysis therefore requires also phosphodiesterase 3B and would occur largely through attenuation of CAMP-stimulated events such as perilipin and hormone-sensitive lipase phosphorylation. The second effect of insulin within the white adipose tissue would be the stimulation of glucose transport. Although the white adipose tissue only accounts for a small fraction of whole body glucose disposal, insulin would stimulate glucose uptake through phosphoinositide 3 kinase dependent and PI3K independent pathways. Insulin-stimulated glucose uptake in the adipocyte, as in muscle, is therefore critically dependent on the IRS1 PI3K AKT axis. Now that we have gone through the effects of insulin in the three major organs that are sensitive to concentrations of blood sugar, we now move towards the final fate of insulin. Insulin must be cleared from the general circulation. So we end tracing the journey of insulin with a brief review of its removal from the system. Insulin is cleared mainly in two organs, the liver and the kidneys, with the former contributing to a greater extent than the latter, removing endogenously released hormone. Skeletal muscle, adipocytes, and other extrahepatic insulin target tissues or cells may contribute to peripheral insulin clearance. Following its pulsatile secretion from pancreatic beta cells, insulin is transported rapidly via the portal vein to reach the hepatocytes by crossing the fenestrations in the liver sinusoidal epithelium. Under normal physiological conditions, up to 80% of secreted insulin is cleared during its first passage through the liver. Upon its secretion from the pancreatic beta cells, insulin reaches the liver through the portal circulation to exert its action and eventually undergo clearance in the hepatocytes. In addition to insulin secretion, hepatic insulin clearance regulates the homeostatic level of insulin required to reach peripheral insulin tissues to elicit proper insulin action. Receptor-mediated insulin uptake, followed by its degradation, constitutes the basic mechanism of insulin clearance. Upon its phosphorylation by the insulin receptor by tyrosine kinase, carcinoembryonic antigen-related cell adhesion molecule 1, or CCAM1, takes part in the insulin-insulin receptor complex to increase the rate of its endocytosis and targeting to the degradation pathways. CCAM1 is a transmembrane glycoprotein that belongs to the immunoglobulin superfamily of the CEA family of cell adhesion molecules. Insulin clearance in hepatocytes is therefore initiated by activation of the insulin receptor by acute release of insulin pulses from the beta cells, which in turn phosphorylates CCAM to mediate insulin targeting to its degradation process. Receptor-mediated uptake followed by insulin degradation by hepatocytes constitutes the basic mechanism of hepatic insulin clearance. Unprocessed insulin is delivered into the systemic circulation to exert its action in target peripheral tissues and brain, and to undergo further receptor-mediated uptake and degradation in the periphery. Whereas insulin undergoes degradation, the insulin receptor may either recycle back to the plasma membrane or translocate into lysosomes for degradation. Clearance from the circulation is the final fate of the insulin molecule. We have gone through the journey of insulin from the time the chromosome 11 gene has been replicated, transcribed, and translated in the pancreatic beta cell to its storage within the same cell, its secretion, and its activation of glucose metabolic pathways or other energy pathways in three key organs. We have finally described the biochemical and physiological events covering all those necessary parts of the journey of insulin across the body. In conclusion, insulin is a key metabolic hormone, and defects in production, secretion, and viability of pancreatic beta cells lead to diabetes mellitus, a disease of epidemic proportions. Insulin production is regulated at various stages in a coordinated and integrated manner. Insulin production and secretion pathways are responsive to or sensitive to nutrient levels, as well as to hormones and other environmental factors. With secretion in healthy individuals, fine-tuned to match the metabolic needs of the body. This presentation has been meaningful as it coincides with the centennial of insulin discovery. Now, patients can have a normal lifespan. But as Banting said in his Nobel lecture, insulin is not a cure. It is a treatment that keeps people alive. There has been a lot of development since 100 years back. And as you can see from this timeline, there are so many new information and technology in relation to the first insulin discovery. If we are to prevent the long-term complications of the disease, we need to understand how insulin is normally secreted and what goes wrong with this process in diabetes. Again, my appreciation to the whole society for allowing me to join you in this virtual series of select lectures and allowing me as well to share with you new updates on the biochemical insulin action. And for more information in relation to the content of this presentation, I refer you to the references that were used for the content of my lecture. Again, thank you and a pleasant evening to everyone.
Wow. Wow, congratulations po ma'am and thank you very much. Palakpakan po natin si Doktora. Yung parang inaral natin ng ilang buwan. Pinagsama-sama ni ma'am sa
isang oras.
Isang oras and probably less. So I would like, there are some questions in the chat box, ma'am. I would like though to ask the first. We go on fasting, no? Like for us Muslims in times of Ramadan, and now it's also very common for some people to go on prolonged fasting. So what would be the effect on the insulin secretion and release during this time?
Okay, that's a very interesting question. What I just described, in fact, is what happens in the normal pattern of dietary intake, like we have breakfast, lunch, and dinner, because that is what the body is really prepared for. And biologically, that's how we respond. Okay? So in cases where the cell, the pancreatic cell, is stimulated by variation in diet, as mentioned earlier, in nutrient intake, it will have to adjust in terms of the insulin synthesis as well as secretion. And over time, studies have shown that over time, because of this adjustment, insulin has maintained a particular plasticity, if you may call it like that. It's that similar to the plasticity of the neurons that will allow it to adapt to prolonged changes in dietary intake. Okay, and it will vary its reactions to the nutrients that it will that the body will be made that will be exposed to. I hope that answers the question. Or
yes. In connection with that, Dr. isabay ko na yung sunod ko na yung tanong din ni Dr. Elwin, no? Ang tanong naman po is the effect of the ketogenic diet on insulin secretion, insulin action.
Right. If I remember right, we had a Dr. Ang and I were also invited to the REF, right? That was pre-pandemic, and Dr. Ang and I were assigned to discuss the effect of different types of diets. Tama? And I think Dr. Ang should be the best person to answer that because it was his topic. But anyway,
Maybe
but anyway, yeah, from my end, kasi.
Can you
but
join
from my end,
well, Dr.
Yes, ma'am.
Yes, ma'am.
Kasi from what in relation to what I just discussed, ketogenic diet is basically that, based on a specific type of nutrient, which are ketone bodies. Okay, so we are bombarding the body with ketone bodies in the hope that the other nutrients are going to be utilized by the cells. That's basically the concept related to the variation of the nutrients. Okay, and in that, again, similar to what I just mentioned, over time there's a certain kind of the pancreatic cells being attuned to the changes, okay, of the diet for a prolonged period of time. Okay? And so they will have to adjust their own synthesis and secretion of insulin. Tama ba yun, Dr. Ang? O baka mas maganda yung explanation mo?
I heard my name. I thought I'm finished.
Oh, kasi topic mo yan nun
Yes,
sa
we will-
REF yung ketogenic diet eh.
Dr. Ang.
Now,
Ikaw,
you can recall the fat diet that we discussed and presented last time was for weight loss. Not
yeah.
about diabetes or insulin action or insulin secretion. But it's really difficult to quantify. If you ask what would be the effect of ketogenic diet in terms of insulin secretion as well as insulin action, those are totally different things. No? And if you really stick on what is ketogenic diet, because ketogenic diet is very, very low carbohydrates diet.
Correct.
5%. No? And there are trials in type 2 diabetes. The outcome that they want is to lose weight. So they're trying to find out if ketogenic and the other fat diet was intermittent fasting. This type 2 this overweight will lose weight. So they are trying to look at efficacy as far as the weight loss. Okay. They are not looking at the insulin sensitivity or whatever.
Yes.
However, ketogenic diet with a low carbohydrates and a intermittent fasting, if the patient is taking insulin or SU, they have to reduce their dose because for fear of hypoglycemia in these trials. So in these few trials, I think 3 or 4 trials, they were able to prove that this ketogenic as well as intermittent fasting are effective as far as weight loss. Okay. Now I think there's a conflicting studies on animal regarding ketogenic because we don't know how much ketogenic and how long do you give ketogenic diet or how long would you receive the ketogenic diet. Because if you go on with ketogenic diet, you will lose weight. And losing weight itself will improve your insulin resistance. Okay, you improve your insulin sensitivity. However, there are animal studies to show that when you undergo ketogenic diet, you develop hepatic insulin resistance.
Yes.
And eventually, the byproduct of DAG or diglycerol acid, the diacylglycerol and then ceramide will be accumulated, and then you would have some insulin resistance in the liver. That's all I know. But there are some animal studies that do not agree on this. So I think it depends on how low is the keto, how high is the keto, how low is the carbohydrates, and what type of study. So in other words, I'm finished.
Yeah. But in the normal reaction of the body, or at least the key organs like the adipose tissue, when there is a low level of glucose, then the uptake is definitely going to be diminished, or it's going to be inhibited. And therefore, the cells will have to adjust also in terms of their nutritional needs. This is for healthy, normal individuals. Okay, for diabetics, it would be a different picture. Okay, and the ketogenic diet is basically doing that. It's trying to let the body utilize the
the stored nutrients, and in this particular case, it has to be glycogen, which can only be available for 12 hours. Okay, and after which, the body will now have to shift to the fatty acid stores, okay, which are first in the adipose tissues as triglycerides. So immobilize, there will be mobilization of that until such that the body will reach a certain point that there are no longer the
Genesis
amount.
naman.
Yeah.
Shift na sa group.
Right, but this is under conditions of normal individuals. For diabetics, it would be far different because we lose the sensing mechanisms as well as the effect of insulin.
Okay, thank you po, sir. Thank you, ma'am. Another question po is about the is there a direct effect of your insulin in the brain metabolism? Kasi kanina sabi natin sa muscles.
Yeah. Yeah, I did not
brain.
include. I did not include in the presentation. There are actually two effects of insulin. They are classified as direct effect, which are the ones that I just presented, and you have the indirect effects. And those indirect effects will also be observed in other tissues, that is particularly the brain, that would require insulin as its major source of energy. Okay, so when we talk about indirect effect, it is not through the mechanism of your insulin receptor binding. Kaya nga indirect eh. It's going to stimulate other enzymes that will allow these key organs, the vital organs that don't have the insulin receptors, to still be able to utilize the glucose that they need. So that's what we mean by effect of insulin, not directly, but it has an indirect effect. Okay.
Okay, Dr. Fatma's back. Nakamute ka, Teg.
Nawala si Fatma, oh.
Ayan, nakamute ka. Nakamute ka pa.
Okay.
Yeah.
Sorry, I was lost. Signal is not so good here in Zamboanga City. But it's still nice to be able to join you all, Doctora, from this very far end of the country. Ay, nabasa na yata ni Danny yung isang tanong about
The brain. Sige, yung next na.
Ikaw na magbasa ng kasunod na tanong. Meron pa ba, Danny?
Sige, may ma'am, mga mineral deficiencies daw, calcium, mga minerals.
Yeah.
Deficiencies, lalo na yung chromium, ma'am. Diba?
Right.
May mga capsule. Ano naman daw po yung effect niya sa insulin secretion and action?
Right. In metabolism of nutrients, in energy metabolism, it is always important to consider the role of your micronutrients, among which would be your magnesium, okay, your magnesium, manganese, because these are needed to activate certain enzymes that will convert the nutrients into the desired metabolic product that will eventually produce ATP. Okay, so in the absence of this, or in the deficiency of these key micronutrients, then definitely there is going to be an effect on how the energy is going to be utilized by the cell, not directly on insulin, but on the metabolic pathways. So that would be the role of these nutrients. Now, the more important nutrients as far as insulin is concerned is zinc. Okay, because zinc is needed to form the mature form of insulin in the beta cells. Remember, in the storage of insulin in the granules, the form that is stored is going to be a hexamer that is linked to zinc. So in the absence or low levels of zinc, then that part of the synthesis of the hormone is also going to be compromised. So yun, yung zinc ang pinaka-importante doon sa synthesis ng enzyme, ng hormone.
So kaya pala yung mga ini-inject endogenous insulin.
Since we don't monitor our insulin zinc regularly. Wala namang test kami dito sa I don't know if there Manila. So will it be safe? Will it not do good? Will it do good to routinely give zinc, Doctora, for our patients we're giving?
Ako, I will have to say that I am not confident to answer that because I don't practice diabetes medicine. No? So maybe I can ask Dr. Ang, who is more on the position to do that.
We don't
we don't have any strong evidence in taking extra trace element like zinc that will improve diabetes. They're also leveling chromium as a adjunct treatment to diabetes. Again, we don't have that large, huge data to say it's just
Very-
like ivermectin for COVID-19.
Talaga, i-mention talaga yun ni Dr. Ang.
They try to prevent physician to prescribe ivermectin other than clinical trial because there's a lot since several months or since this year, there are a lot of side effects from ivermectin that were documented because we do not know what is the dose that will be effective, the dose for the side that would give you side effects, etc. And we don't even know if it's really effective for COVID-19. Be aware of the mu variant. We have already 12 variant. Bakit ba tayo nasa COVID? Let's have
Sir, pinasok mo si ivermectin eh.
Let's zinc.
Answer the Can you explain insulitis? What happened among type 1 patient? Okay, can I answer?
Clinically answer.
For type 1, okay? Unless ma'am would like to answer
Sir, ikaw na. Mas magaling ka dyan. No?
Okay, now in the pathogenesis of type 1, it's stated there that it is a cellularly mediated autoimmune disease.
Right.
So in our immune system, there are two types. There are cellular and hormonal. Okay, so when you say cellular, you would see the T cells, the killer cells, the T cells and macrophages that destroying the beta cells. Okay. However, you have the hormonal, like anti-God, anti-insulin, etc. But these are not directly destroying the beta cells. These are markers that there are something going on the beta cells of type 1, the cellular mediated that are destroying the beta cells. So in other words, the insulitis, the cellular is the fire, and the smoke is the the hormonal, the anti-God, etc. The anti-insulin, the anti-God. These are the markers. No? These are the smoke, and the fire is the cellular mediated. So that's why you have insulitis in type 1 that you don't see in type 2 diabetes because it's immune mediated,
autoimmune.
Immune mediated autoimmune mediated
autoimmune disease. Yeah.
Meaning the T cells, macrophages, etc. are destroying the beta cells, not the antibodies.
Correct.
Thank you, Dr. Ang. For all our attendees tonight, yung present ni Doktora will be a very nice parang like a book to keep, and also that of Dr. Ang. Again, we in PSD can give you a copy of that so you can review. Siyempre, pag biochemistry, parang isang pasada, ma'am, parang hindi pa natin yan magandang balikan, pakinggan ulit, and then that will help us understand it better. So, do we still have any other questions from the chat group?
Meron na lang isa po.
Meron pa bang isa? Basahin mo na dapat.
From Dr. Toti po yung about the importance of pulsatile or oscillatory nature of insulin secretion. Katulad yung sabi niyo, yung first phase tsaka yung
Yeah.
second phase. Kasi nawawala po ito sa mga type 2 diabetic patient.
Correct. Oo. Oh, that's very important kasi 'di ba the reserve pool there are more insulin stored in the reserve pool. So the first phase of the secretion process is very short and also with only a small amount of insulin being secreted. So to ensure that more insulin is going to be made available to the other cells, the reserve pool must be there should be a mechanism to allow for the reserve pool to be mobilized. And because dadaan siya sa napakaraming pathway, no, sa cortical actin, all these cellular proteins na dadaanan niya. If that is working well, then it will allow the reserve pool to replenish the readily releasable pool. So that's a significance. Kung hindi ma-replenish yung first pool, then that means that we will have insufficient insulin. And that will lead to pathologies.
So, you mentioned a while ago, ma'am, dun sa biphasic insulin secretion, yung exocytosis, yung release ng second is positively related. Yun ba yun? To the first phase. So, ibig sabihin, maba na yung first phase, ang second phase mo, bababa na rin. Can we, parang ganun ba yun?
No. No, pag bumaba yung first phase, 'di ba? Ano lang 'yon? A brief release of insulin, okay? And then madideplete na 'yon yung readily releasable pool. So to make sure that there is still available insulin kasi that pool is not sufficient to be distributed to the key organs. So what the body should the pancreas should do would be to recruit more that are stored in the reserve pool. So that will now replace what was released initially during the first phase. So hindi yun yung pag bumaba na yung unang pool, titigil na rin yung isa. In fact, it's not. It's going to stimulate the reserve pool for its mobilization towards the plasma membrane for its own release.
Okay. So, yan yung natin sa basal bolus kasi na
And si Kuya, naka-raise ang kamay ni Kuya, oh.
Yeah.
Yes, I think Dr. Hipolito is also her question also pertains to another character of insulin secretion, which is pulsatile and oscillatory, which is different from first phase and second phase. Yung pulsatile or oscillatory is that even during the first phase, it is not very smooth up and down. It is oscillating up and down little by little. I think that is what Dr. Hipolito is referring to. And that is important also.
Yeah.
Yes, even though we did not know its value before, I think there are studies showing that it improves insulin action and diminishes insulin resistance.
Resistance, Thank you, Kuya.
Hindi lang isang beses lalabas kasi, di ba, yung naka-store, ma'am. Meron pang iba. So, kaya sunod-sunod din. Siguro ako magtanong din. Yung mga insulin analog natin, will it have the same journey as the human insulin?
What I described is endogenous. Depende, si Dr. Ang siguro, kasi yung topic niya about insulin he might be able to elucidate on that.
Lalo na kung may mga problema sa liver.
Correct.
Will it have the same journey?
Well, what was the question? Just to remind the student, okay. Now, the first phase and second phase insulin secretion, the pattern, this you cannot see when you give it when you give the glucose orally. No?
Yeah.
This is in a laboratory wherein you give IV insulin.
Hyperglycemic camp. Yeah.
hyperglycemic clamp, you give IV glucose, so that you will see the pattern on the first phase and second phase insulin. Okay. So the question was, is
Kapag ano daw, sir,
insulin
yung mga ini-inject na
Muscles.
insulin, it has the same pattern.
of what?
The biphasic, sir.
Yung
Yung journey daw ng insulin, sir, will it go through the same journey as the human insulin? Yung mga analog.
It depends on the analog. When you talk about insulin analog, that is rapid, no? Rapid, I mean, yung much quicker, like glulysin, aspart. These are only changes in the certain amino acids that will make the insulin crystal non-hexameric, either dimeric, monomeric. That's why the mechanism here, the technology, they change the amino acid like lysine. They remove the 20. They change the 28 and 29, invert it
Amino acids.
of the B chain 28, 29. They change the amino acid and become lysine. Actually, it's proline and lysine. They become lysine and proline. So in doing so, it becomes dimeric or monomeric. And then, you know, if you inject it in the parenteral, instead of hexamer, dimer or monomeric, the absorption is faster. So when it is absorbed, the journey is, I think, is the same. The only difference is the onset of action.
Yeah.
It's the same. They just change it's an analog, it changed the amino acid sequence so that it won't form an hexamer. Because the delay is the hexamer. It has to be broken down to dimer and monomer before it is absorbed in the fat, no? In the fat depot, in the parenterally, into the circulation. So you limit, you reduce the duration of absorption. Okay. So I think it's the same because when it is absorbed, it's the same. In fact, using rapid insulin and regular insulin IV, there's not much advantage. The only difference is more expensive. They did a study on this. They found out that there are slight differences, but it's all the same. The effect, the pharmacokinetic, pharmacodynamic, the pharmacodynamics is the same, very similar, because you bypass the parenteral, you give it IV. And it all depends on what type of analog. Is it basal analog? Basal analog, it protract or prolong by changing. There are a lot of technology in prolonging the insulin absorption in the parenteral. Like glargine, they change against the B30 position, and then they put some zinc. Kaya nga pH yung glargine medyo low pH, medyo acidic yun eh. While lebermir, they put an fatty acid so that when it is absorbed, it will binds with albumin. Okay, so when it binds with albumin, it makes the half-life longer. I think the fatty acid is myristic acid. There's already once weekly insulin. It's not yet available here, also in the state, but they are developing it. It is it's a basal insulin like glargine, and it's a fusion protein combined with the FC portion, no? Immunoglobulin FC. So when you combine it, it's more stable. The half-life becomes longer. Okay, that just like Trulicity. Trulicity dulaglutide use technology for it to be once weekly. Okay, so I think after the degradation, after the absorption, I think it's the same pathway. No?
Thank you, Dr. For clearly answering. No? Danny, okay na ba sa'yo? Nasagot na ni Dr. Ang. So I think our last question for tonight. It's always very interesting to talk about insulin because yan ang natin sa ating practice. No? So again, it was really honor, a privilege to listen to you tonight. Dr. Villamor, thank you so much for joining us, and also Dr. Ang. Again, to our attendees, we will try to give you the copy of the lectures, just like what we do in the past, so you can review, you can study them again. I myself will go through that siguro, Doktora, mga 3 times more than
Kahit na
I
aral na
kept
natin,
going
no?
back.
Yes.
And I finished BS Chemistry, so it's like going back there. So, again, to our attendees, maraming salamat po for joining us. Before we leave, we would like to invite you to our 30th anniversary celebration. This will be on October 2, 2021. Please mark your calendar. We will have a very fun and exciting fellowship. Don't miss. We're giving away lots of exciting prizes from Dr. Danny Baltimore. So, from 5
From Zamboanga yan, ha?
to
Mamimigay ng
galing sa
perla si Doktora.
po yun to 9 p.m. I-block nyo na po yung inyong schedule, and let's all get together. It will be our annual convention and also our celebration of our 30th year for the Philippine Society of Diabetologists. So again, with that, this is your host and your moderator for tonight, Dr. Fatma Q, saying maraming salamat po sa inyong lahat.
Thank you.
Picture naman
Doktora,
tayo.
picture taking po.
Ay, picture daw.
Okay, open. Thank you po sa Sanofi din.
Pa-open din natin kung sinong nandyan. Maraming salamat sa Sanofi. Maraming salamat din sa ating technical team led by Gets. And sila Sir, sino pa nandyan, Jay. And si Ma'am Tin.
Tsaka si Ma'am Tin.
And Ma'am Tin, maraming salamat.
Sanofi, thank you po. Picture.
Sanofi. The
Open
picture,
na
web.
lang, ma'am.
Camera.
Yeah.